Hi, this is Kelsey Tuckett. Just on here on class and just here to share what I learned from the lesson one group problems. I just wanted to go through some of the things that I came to know and just wanted to know if, there's anyone out there that wants to help me or that I did good. I just wanted to let it out there. So just going through the questions took me a little time, but I figured out that I could just list everything out just as if I was doing Punnett squares for each individual trait. And then just going through and exploring, what combinations can be made as if I was doing an actual Punnett square and then taking all those and putting them in ratios. So that way I could do the further questions where it asks about proportions of the offspring. Went through and just figured out that part A for question one was 128th of a expected genotype for that question. And then part B, I got 256th. Can't say these, so forgive me. I got 0 because the little d's, the word escapes me, but because those aren't existent in the combinations, because we have a big D and a little d, that it's impossible to have two little d's, thus just canceling out everything else. And then for the last part D, I got 1/32 for the that would be expected for that genotype. In the blanks, the paired homologous chromosomes are sometimes called, I figured bivalents or tetrads, depending on what they are. But I have a feeling that other people know a little bit better. Part B of question 2, right? Yeah, question 2. The structure that connects this centromere to the microtubules of the meiotic spindle is called a spindle poles. Because I figured, going through the reading again, just looking over what it said, that seemed to be what fit that answer best, because there's at the end that connect to the genes, if I remember correctly. I could totally be misquoting this. A diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 8 sister chromatids, while a fruit fly cell in metaphase 2 has 16 sister chromatids. So I just took the fact that it had 4 pairs of chromosomes, and sister chromatids are part of the chromosome. So there's two sister chromatids to one chromosome, and so I just multiplied by 4 to get 8. And then in metaphase 2, it splits into 2 different cells. Took that and then did the same thing, multiplied it, because now there's multiples. For part D, monohybrid cross is a type of genetic cross that can determine if a purple flowered pea plant has the homozygous or heterozygous pea genotype. And so going with the monohybrid cross, just going back to like high school thinking of doing all those crosses that there's only looking for one trait, so that's what that is. Question E, a non-functional allele is sometimes called a null. I think that's just the casual term for it in the reading. A null allele, functional allele, is called a gain-of-function allele. So hopefully those are appropriate answers. I was looking through some of the other ones and noticed that there were some different ones. So if anyone has any answers for that one, that can help me clarify. And then Mendel's law of independent involves 2 traits, while Mendel's law of segregation involves a single trait. And that one just seemed very straightforward. So question 3 kind of confused me, so I'm not quite sure what I did. I drew a diagram. That's what the colored portion is right here. This part. Because I decided to just color code everything, show that how everything was set up for when everything's in metaphase 1 in this particular homozygous chromosome. And then for part B, so all of this in this section is part of part B in the end, because that's where I got these answers. With just how there would be some genotypes. But I have a feeling I needed to write out more. So, lessons learned. Part C, talking about the same thing. Let me go back to that main question. Says that suppose an organism has a genotype T capital R lowercase r lowercase y, and then assume the alleles assort independently. Going through that, how many sister chromatids are found per cell in the organism during metaphase 1? And I figured because of the example that because these are all part of that, and considering that at the particular part in metaphase, maybe I got that one wrong. The process was taking 2, 3, 4, 5, 6, but I think I may have accidentally multiplied it without thinking about it. So I'll have to think about that one again. Part D, though, metaphase 2, because now they're talking about chromosomes, which are that pairing. Each cell in metaphase 2, there are 4 chromosomes during metaphase 2. So I will also have to look over that one, because that one I don't think is right either, but I could be wrong. Moving on to question 4, because I've been talking for a while now, where it's talking about a horse has 32 pairs of chromosomes. For part A, it talks about how many chromosomes are present at the conclusion of meiosis 1. And so I figured that there would be 16 chromosomes per cell. You'd be splitting that amount of chromosomes in half and preparing for the next step. And so how many tetrads are present per horse cell during prophase 1 of meiosis? There'd be 32 pairs because the crossing over not affecting the sum total. And plus it's the beginning of meiosis. Part C, how many sister chromatids are present in a horse cell during prophase 1 of meiosis? There would be 64. So I just took the original 32 and then multiplied it. That was because I was trying to talk about chromatids. There we go. Brain's back on track. So because there's 32 pairs, each individual chromatid, thus timesing by 2, is 64. There we go. Part D, how many sister chromatids are present per horse cell during metaphase 2 of meiosis? And in that case, there'd be 32 because that's after they're getting ready to split. And so 32, because you've already, with meiosis 1, you've already apart. So now you're so many in each cell. Oh, pardon me. Apparently I'm boring myself. How many bivalents are present per horse cell during anaphase 2 of meiosis? I think 0 would be present because during anaphase 2, the bivalent, I'm just reading this off, would be splitting into haploid cells. So it's just getting prepped to split. So there really wouldn't be anything if it went well. That would be, because bivalents, if I remember correctly, are the pairings because of that bi, meaning 2. That's what that goes with. Question 5. Which is the last question, talks about the tetrahybrid cross in pea plants. And so it explains all of that. And then, so what I did is I went towards the bottom, and you'll have to wait for this to load. or at least I'm having to. I don't know if it's the same. And so there it is right now. I'll scroll back up here. What portion of the offspring will be a tall, round, green, and terminal? So I put everything out. I just took everything in this section. There's my mouse. I took all of this and this because we're taking two of the same cells, that homozygous, and just multiplied or did my little pairing to see what all the fractions would be for each thing. And then went in and figured out the math. So this is for that first problem for a tall, round, green terminal plant. And so it came out being 1/8 for tall and 1/8 for the round, and green was a half in terminal, because when you come up here, for example, the tall, the T's up here, you have this one will produce a tall plant because it has that, in the description of the question, it talks about the capital T being the dominant trait, being tall. And so this one would do it along with the 1/2 trait right here, because that lowercase t is not, it's a recessive trait, so it's not displayed. So then that's how you get the 1/8, is you just multiply them across because you're looking for the probability of it occurring. And so that occurred for many of the next questions, where I'd go through and I had to figure out where each trait would be and what would occur. So just to go through this quickly, questions. Question A was 1/16 of the offspring will meet that criteria of the tall, round, green terminal plant. Question B, what proportion of the offspring plant has that particular genotype displayed on the question? And so I said 1/32 of the offspring will have that genotype. Question C asks for proportion of the offspring will be heterozygous, so that different alleles, so not being the same, not being like both lowercase or both uppercase. And so in that case, there was 16th would have that combination for genes, which is really nice because it's displayed right here. I just went through and wrote them. There'll be 1/64 of the offspring will meet the criteria of being a dwarfed, wrinkled, yellow, and axial pea plant. And so hopefully this I explained what I was thinking well, and you're able to understand it. And I hope that you're able to enjoy this time that we're learning together. And I hope you have a great day. Thank you.